Welcome to the problem-solving set on the genetic code. Problem 1. In the ACS allele of the human actin gene, the second amino acid is changed from lysine to threonine. In the ACB allele, the second amino acid is changed to glutamine. Order these two mutations within the map of the actin gene. Okay, so first let's look at what the wild-type second amino acid is. So, as they say here, the second amino acid is wild-type lysine, right? And the ACS and the ACB are two different mutations. So let's look at what the codon is for lysine normally. So if you look at the genetic code chart, the amino acid codon chart, let's look for lysine. So lysine is down here. The codon AAA or the codon AAG will code for lysine, right? So we'll write that here. So lysine is AAA or AAG. Now in the ACS mutation, this amino acid is changed to a threonine. So let's find the amino acid codon for that. So threonine is right here. It could be ACU, ACC, ACA, or ACG. So it's basically AC and a dash because the third triplet in the codon can be any base. So we'll write AC dash for threonine. And then the ACB mutation changes it to glutamine. So glutamine is right here. It could be CAA or CAG. And this is for glutamine. Okay. So now it wants us to order these mutations within the map. So let's look at what changed from the mutation to make it from the wild-type lysine to whatever amino acid it changed to. So in the ACS mutation, threonine is coded by AC dash, right? Any third base, which means that this second nucleotide here is what changed, right? Because it changed to a C for threonine. So this arrow, we'll call this ACS. And then for the ACB mutation, you can see that it's either CAA or CAG, which changed this first base of the triplet codon, changed to a C. So this is the ACB mutation. So when we're ordering these two mutations within the actin gene, remember that the mRNA that's translated into a protein goes from 5' to 3'. And the first mutation that you run into is ACB mutation, which changed the A to a C. And then you run into the ACS mutation in the next base. Problem 2. A portion of a gene in which a scientist combined one addition and one deletion mutation is shown below. Both wild-type and mutant coding strands are pictured. The mutant yields a wild-type phenotype. Part A. Label the addition and deletion mutations in the DNA. So in order to do that, we're going to compare these two strands and see where the mutant coding strand is different from wild-type. So from 5' to 3', we see that they both start out as CCC, and then the wild type goes on for TCT as the next codon, but the mutant is CTT. Now, if you look through, let's just assume that this T is the one that's deleted. So if you continue along, you'll see it's CTT, which is this here, ATC, which is this here, AAT, that here, and then it's just AC, which is this here, and you're missing this T right here. So this is the deletion. They deleted that thymine, which caused a frame shift, right, over one nucleotide. And then they added this T here, so let's write the addition here. When they added that thymine in, it reverted the reading frame back, which is why this A and this C line up. Part B. Compare the alterations to the amino acid sequence in the mutant from the wild type. So in order to do that, we have to recognize that they're giving us the coding strands, right? And the coding strands in DNA are the ones that are going to be analogous to the mRNA that you make, except that the thymines are going to be changed to uracils. So let's write over the wild type DNA mRNA sequence that we're going to get. So it's going to be 5' CCC UCU, remember because thymines are changed to uracils, UAU, CAA, and UAC. Now let's look, using our genetic code, what these codons actually code for in amino acid language. So the first codon that we're going to look for is CCC. So when you look here, you start on the left-hand column, which is the first base, and you go to C. Then you go over to the second base is C, and again we're looking for CCC, which is right here, and that's a proline. So we write that as PRO, and that's a 3-letter abbreviation for proline. Now the next codon is UCU. So you start at U, you go over to C, and you see that UCU encodes for serine. And the abbreviation for serine is SER. Then you have UAU, which is tyrosine, and that is TYR. Then you have CAA, which is right here, and that's glutamine, which is GLN. And then finally you have UAC, and that again encodes for tyrosine. So this is the wild type amino acid sequence. Now let's do the mutant. So again, we're going to rewrite our coding strand DNA as an mRNA sequence. So it's going to start CCC, then CUU, AUC, AAU, and UAC. So again, from beginning, we know that CCC encodes for proline. And now let's look for CUU. If you go across, CUU encodes for leucine. Then AUC encodes for isoleucine, which is ILE. Then you have AAU, which goes for asparagine, which is ASN. And then finally, UAC is the same as above, and that encodes for tyrosine. So these are the amino acid sequence. This is the wild type right here, and this is the mutant. So you see they both start out with proline, but then on the top you go serine, tyrosine, glutamine, and then on the bottom you go leucine, isoleucine, and asparagine, and then they both end with tyrosine. So that is part B. Now for part C, it says, briefly explain how a change in amino acids can still yield a wild type phenotype, because remember, on the top here it said that the mutant yields a wild type phenotype. Well, this is just an example, and the actual changes in the amino acid sequence, this primary sequence, may not actually fit this explanation. But the general answer is that amino acids are grouped into different classes with similar properties. So you have the nonpolar amino acids, you have polar amino acids that are not charged, and then you have the charged amino acids, which could be acidic or basic. And all those properties depend on the side chains of the amino acids. And if you switch, let's say, one acidic amino acid for another acidic amino acid, or you change one hydrophilic or nonpolar for another, then they'll still react similarly, and the protein may still fold in the same general pattern. So when the proteins fold in the same way, that means they have the same 3D structure, which corresponds to their function. So again, this is a general explanation, but please note that serine to leucine, tyrosine to isoleucine, these don't necessarily fit this general explanation, and they might not be in the same property group, but that's a way to explain how this change in amino acid can still yield a wild type phenotype, because the protein is still going to fold with the same characteristics. Problem 3. The amino acid sequence of a part of the myosin protein has been determined to be glycine, alanine, proline, arginine, and lysine, and after treatment with a mutagen known to cause frame shift mutations, the mutant protein is purified and its amino acid sequence is determined to be glycine, histidine, glutamine, glycine, and lysine. It says determine the sequence of nucleotides encoding the mutant allele of this myosin derivative. Okay, so what they're telling us is that they have a wild-type amino acid sequence, and then after treatment with a mutagen that can cause frame shift mutations in the DNA, you get a new protein that has a different amino acid sequence. So what we're going to do, we're going to label this wild-type, and then label this mutant, and we're going to determine what the mRNA sequence would be for these amino acid sequences and then compare them. Okay, so let's do the wild type first. So the first amino acid is glycine. So we're going to go to the genetic code chart and look for glycine. So glycines are down here, and they can be encoded by GG and then UC, A, or G. So we're going to write this as GG dash. Okay, now the next one is alanine. So when you're looking for alanine, you see that they're down here. Let me just make sure. You have to be careful when using this genetic code in this type of way because some amino acids can be encoded by at least 6 different codons. So you definitely want to be careful. So alanine, it looks like these are the only 4. So we're going to write that as GC dash. Okay, then proline. See it up here. Looks like those are the only 4, so we're going to write that as CC dash. And then arginine. Let's see. So we have 2 arginines down here. And then we also have 4 arginines up here. So we're going to write this as either CG dash, so we'll write that first. But then it could also be AGA or AGG. So I'm going to write that right here, my AGA, AGG. Okay, and then lysine. See, lysine. Okay, lysine's right here. So it's either AAA or AAG. Okay, so this is, I know it's missing a lot, but once we compare it with the mutant sequence, we'll be able to fill this in, okay? So now let's look at the mutant sequence. So again, the first amino acid is glycine, so that's the same as above. So we're going to write GG dash. Then the next one is histidine. So histidine, CAU, CAC. Let me just double check that it's not anywhere else. Okay, so it's just CAU or CAC. Okay, then the next one is glutamine. And now, if your professor wants you to know the 3-letter abbreviation for amino acids, just remember that GLN is glutamine and GLU is glutamate or glutamic acid. Okay, so now we're looking for glutamine with an N in it. So it's not glutamate, it's glutamine. And you're looking through glutamine, we see either CAA or CAG. Okay, now the next one is glycine. So again, that's down here. It's GG dash. And then lysine is the same as above, so we're going to write AAA or AAG. Okay, now we're going to compare these. So if you look, the first amino acid in both the wild type and the mutant sequence are the same, right? They're both glycine. So what we're going to do, we're just going to ignore these for now. Now, if you look, the nucleotide sequence in the wild type is GC dash, and the nucleotide sequence in the mutant is CAU. So what do you think happened here? It's plausible that this G was deleted by this mutagen, which would frame shift the whole sequence over one, right? So this C matches with this, and then because this is a dash here, it means that it could definitely be an A, so that matches there. Then you have a C next, so that matches with this one. So we know that this is the codon that's actually there, and that's because there's no U to match with the wild type. Then you have C matches. Then again, a dash can definitely be the A. And then, in the wild type sequence, remember that arginine can be encoded by 6 different codons, right? So those codons either begin with a C or they begin with an A. Now the mutant nucleotide sequence there has either an A or a G, so we know that it's going to be one of these A's that match up here. So therefore this codon, that's not the codon that encodes for arginine in that case. Then the next nucleotide is G, matches here. And then there's a G in the mutant, so it must be this nucleotide. So that's the codon that was in the wild type. And then you have a dash, which means that the A can definitely fit there. Then you have an A again. And then you have either an A or a G in the wild type and an A or an A in the mutant. So we know that it's this A that is matching. So this was the codon and not this one. And then because we don't know anything further, like we know that because ... means that there is a further sequence, but we don't actually see it. So we don't know if it's the last nucleotide is going to be A or it's going to be G. It could be either because we know that we have lysine there, okay? So now it wants us to determine the sequence of nucleotides encoding this mutant allele. So let's draw this out. So we ignored this first codon because glycine we knew didn't change. So we're going to write that. Remember, this is the mutant. We're just going to write that as G dash, and again, because of the wobble theory, we don't know the identity of that third base, but that's okay, we could just write the dash. Then we knew that the next codon is going to be CAC because the guanine was deleted in the wild type from the mutagen, and then it frame shift over, and then with the matching, we found that the next sequence was CAC, and then we found the next sequence. I didn't cross it out here, but it was actually this sequence, so CAA. Then we found it to be GG, and it matched up with an A, so it's GGA. And then finally we have AA, and then a dash again, so that this dash we will actually write as A/G, because it's not a dash, it can't be any of the 4, it can only be A or G. So this is the sequence of nucleotides that encodes this mutant allele of the myosin derivative. So we know for a fact 13 nucleotides for certain, and 2 we're uncertain of. Okay, so it's a little difficult. You have to get used to the genetic code chart and looking at the codons. Remember that the codons always run from 5' to 3'. And that's how the chart is given. So these codons here are written as they were 5' to 3' on the mRNA. But remember that the anticodons, which are on the tRNA, those run from 3' to 5', and they're the complementary base pairs to this. Okay? Problem 4. How many different nucleotide sequences can encode the following amino acid sequences? So remember, when they write NH2 and COOH at the end of amino acid sequences, this is just convention to write the N terminus and the carboxy or C terminus. So we're not going to consider that when we do these calculations. The way we're going to do this is we're going to look at each amino acid and say how many codons can encode that amino acid, right? Because that would be a different nucleotide sequence. So the first one, the amino acid, is methionine. So as you may know, that is the start codon, and there's only one codon that can encode for that amino acid, which is AUG. So we're going to write 1. Then the next amino acid is alanine. So let's look at how many amino acid codons can encode for this. So when you look through, you find alanine. There are 4. And just to make sure, take a quick glance over to see if there may be another 2, some of them that encode for this amino acid as well. But it looks like these are the only 4. So let's write, we're going to multiply across, because remember, if there's 4 different sequences that can encode for alanine, and then there's 2 that encode for tryptophan, then there's going to be 8. So it's basically another application of the probability rule. So now let's look for tryptophan. Okay, so I see 1 here for tryptophan. And then finally for glycine, they're down here, and there are 4 different codons that can encode for glycine. So when you multiply these across, you get 16, and that means that there's 16 possible nucleotide sequences that are different, and that can encode these same amino acid sequences. And remember, this is really an application of the wobble rule, and that's when you have a codon that goes from 5' to 3', it's a triplet, right? And this third nucleotide can alternate, and a lot of times it doesn't really affect what amino acid it encodes for. So as you can see for like glycine, basically anything that begins with GG can encode for glycine, and for alanine, anything that begins with GC can encode for alanine. So this is an application of the wobble theory. Now for B, the first amino acid is glycine, which we just found before, can be encoded by 4 different codons. Then you have serine. So serine, I see 2 here, 2 different codons. And just a quick look, you can see there's actually another 4 that encode for serine, so total is 6. I think that's the maximum, so I think that there are a few amino acids that can be translated with 6 different codons, but I think that's the maximum number. Then you have tyrosine. So tyrosine, there's 2 up here. And if you scan through, those seem to be the only ones. So there's just 2. And then for leucine, you have, let's see, 1, 2, 3, you have 6 here. And if you scan through, those are the only ones. So there's 6 for leucine again. So when you multiply this across, let's take out the calculator for this one. You have 4 times 6 times 2 times 6, and that equals 288. So there's 288 different possible nucleotide sequences that can encode this amino acid sequence. Now for part C, the first amino acid is proline. So if you look through, you see there are 4 here. Let me just make sure those are the only ones. Okay, so there's just 4 for proline. Then you have histidine. So there are 2 histidines here. And again, those seems like the only ones, so we're going to write 2. Then you have glutamine. Remember, the N is for glutamine. So we have 2 here. And it looks like those are the only ones. Remember that this glutamate here is actually glutamic acid. And this is abbreviated by GLU, whereas this glutamine is GLN. So there's only 2. And then lysine, we have 2 here. And I think those are the only codons, so it's going to be 2 again. So when you multiply that across, you get 32. So there's 32 different nucleotide sequences that can give this amino acid sequence. Problem 5. two separate single base substitutions in the gene for hemoglobin affected the sixth amino acid. In one mutant allele, the sixth amino acid is now histidine. In the other, it was serine. Out of the following choices, what was the most likely amino acid at position 6 in the wild-type hemoglobin? Okay, so this is another question that's going to require us to compare and contrast codons and the nucleotide sequence of those codons. So let's just highlight a few things we want to remember. First of all, the mutants come from two separate single base substitutions, and then one mutant gives a histidine as the sixth amino acid, and the other gives a serine at the sixth amino acid. So we can write M1 for mutation 1 gives histidine, and M2 gives serine. So now let's use our genetic code to determine what codons can encode for these amino acids. First, let's look for histidine. So we see CAU and CAC can both encode for histidine. And if you scan through, you can see that these are the only two. So we're going to write CAU or CAC. And then for serine, we can see that either AGU or AGC can give it, but there's also UCU, UCC, UCA, and UCG. So first let's write UC dash, and then AGU and AGC. Okay, now we're going to do the same for each of the possible answers. So we're going to do for valine, tryptophan, glycine, arginine, and proline. And what we're going to do is compare and say, can one single base pair substitution convert it to these new mutations, okay? So let's look for valine first. Valine can be GU dash, and let me just make sure that those are the only ones. Okay, so valine we're going to write as GU dash. Tryptophan only has one codon, which is UGG. Glycine, you scan through, and we see that glycine can be encoded by these four codons, so we're going to write that as GG dash. Then we have arginine, so arginine can be AGA or AGG, but it can also be these four. So let's first write CG dash, and then also AGA and AGG. And then finally for proline, proline can be encoded by these four. And those look like the only four, so we're going to write that as CC dash. Okay, so now let's start comparing, now that we have everything written down. So if we're looking at valine, valine is GU and then any nucleotide at that third position. So if we want to convert valine to histidine, that means that we would have to mutate this G into a C and mutate this U into an A. Now remember, the problem said that this mutation only caused single base substitutions, so one base was substituted in each of the two mutants. Now because it would require two base substitutions to get from here, we're going to cross that valine because that definitely cannot be the original wild-type amino acid at position 6. Now for tryptophan, we know that the only codon that encodes for tryptophan is UGG. So if we're looking to convert tryptophan to histidine, that means that this uracil would have to be changed to cytosine, this guanine would have to be changed to adenine, And then this guanine would also have to be changed to either uracil or cytosine. So that's 3 base substitutions, so that is definitely not the wild type amino acid. Now for glycine, glycine can be GG dash. So to get from glycine to histidine, you would have to change this G to a C and this G to an A. So again, that's more than one base substitution, so glycine is out. Now arginine. If we're looking to convert arginine into histidine, let's say that arginine was encoded by a CG dash codon. That means that the C can stay the same, and the G would have to be converted into an A, and then the dash could definitely be either U or C. So that works because that's only one base substitution. If arginine was encoded by the AGA or AGG, those wouldn't work because you would have to change the A to a C and then the G to an A. So we're going to cross these out, and we'll circle this as a possibility for histidine, right? Now let's look at proline for histidine. If proline is CC dash, that first C is fine. To convert to histidine, you'd have to change this C to an A, and then the dash could be either U or C. So again, this is only one base substitution, and it is a possibility for histidine. So let's take these two and compare them to the possible serine codons. So if arginine was able to mutate into histidine with only one mutation, that means that the arginine codon would have to be either C, G, U, or C, G, C. And that's because this third codon in histidine has to be either a U or C. So we're going to write that these are the only possibilities because remember, we only have one base substitution to work with here. So if these are the possible amino acids, can any of these be converted with one base substitution into a serine? Well, if you look, if serine was encoded by a UC dash codon, that definitely couldn't work because the C would have to change to a U and the G would have to change to a C, so that would be 2. However, if serine was either encoded by AGU or AGC, then only this first nucleotide would have to be changed. So this works for serine as well. So let's circle arginine because that seems to work for both. Now for proline, let's look for serine. Again, in order to be able to convert from proline to histidine with one base substitution, the proline codon would either have to be CCU or CCC. Now when you're looking for serine, the AGU and the AGC codons would have to be mutated twice, right? 2 bases would have to be substituted in order to get the CCU or CCC. So these would be out for proline. However, for the UC dash serine, that could definitely work because that middle C can change the same. The dash can be either U or C, and only the U would have to be changed from a C. So this is also a possibility for serine. So even though it asks for one answer, there's actually 2 answers within these multiple choices. So either arginine or proline were likely to be the amino acid in the wild type hemoglobin at position 6.